Hi, my name is Elizabeth Mazzio from Florida A&M University, and the title of this work is Metabolic Response to the Mitochondrial Toxin MPP in LDH lactic acid dehydrogenase AB double knockdown LS174T colon cancer cells. So a unique attribute of cancer cells involves a preference for anaerobic glycolysis and the accumulation of lactic acid that occurs even in the presence of high levels of oxygen. Until recently, researchers have proposed this to be a function of maybe a mutation in mitochondria or energy failure. However, we know this is not the case, as the Warburg effect, which is lactic acid, creates a primary role in sustaining the acidity required for tumors to grow, metastasize, and undergo immune escape. We also know this is the case because we can knock out LDH-A and B and have no negative effect on energy. Rather, what we're going to see is greater mitochondrial oxidative phosphorylation and cell respiration. There are studies now also showing that high levels of oxygen and mitochondrial oxidative phosphorylation are actually critical in driving tumorigenic processes, in particular for stem cells. This has set forth a rationale for a new era of research using mitokines, which are mitochondrial targeting and inhibiting drugs. Now, people are trying to put glycolytic inhibitors with mitochondrial inhibitors to create an energy crisis in cancer cells. Will it work? The answer is no. There is no energy crisis. With an elevation of MPP over a dose response in the control vector versus the LDH-AB knockdown, what you see is a rise in lactic acid, rapid use of glucose, sustained level of ATP compensating for reduction of mitochondrial oxygen consumption in the control vector, which means glycolysis is fully functional. And in the LDH-AB knockdown, there's no capacity to produce lactic acid. Very little glucose is being used, but both of these cells have exactly the same or similar amounts of energy, and they're both alive. How can this possibly be? So we backtrack to the control vector versus the LDH-AB double knockdown in the resting cells. And we see high levels of mitochondria. This is right here is Janus green elevation of fluorescence and indicates cell respiration. The control level right here, very small amount of mitochondrial oxygen consumption. The knockdowns are respiring very heavily, and therefore they need more MPP in order to inhibit the electron transport chain. Lastly, we conducted a whole transcriptome analysis of both cell lines with and without MPP. The data is available at NADH GEO 149289. But briefly, MPP had an ability to halt cell respiration as well as some of the transcripts in NADH complex 1. The biggest differential was between the control cells from pyruvate flowing the carbon intermediates to lactate, where the pyruvate was pooling to acetyl coenzyme A in the knockdown. So in conclusion, LDH inhibitors create more of a resistance to mitochondrial toxins than a vulnerability.